So in the Synthetic Forager project, we investigated hierarchical networks. So we iterated the whole scheme and investigated receptive field properties at higher levels, and importantly also at pretty abstract levels in foraging agents, so mobile agents which sampled different types of sensory input. Additionally, we investigated not only the function of sparseness, but actually to have optimal predictability, which means you do not only want to know how the world is structured right now, so for example that there are not only red and green dots, but there are apples and bananas and so on, but you also want to know what's happening if you act in the world. And with this concept of predictability, you utilize the predictability of features to select which features should be processed. This is an important step beyond making predictions in itself. And so far, predictability is an important concept, and actually can be shown that sparse representations, so representations which are sparse right now, as well the prediction or the entropy of the prediction is optimally sparse, have maximal predictability. This is shown here with so-called place fields from a random distribution, which is like a salt and pepper pattern, cutting and merging parts of the receptive fields in order to optimize predictability. You slowly form more compact regions, and actually these demonstrate optimal predictability, which also is a superset of optimal stability. Optimally stable representations are obviously predictable, but not necessarily the other way around. Predictable stimulus might be dynamic as well. This would be on the top of the sensory hierarchy, useful for producing motor behavior, like for example in foraging agents, or in natural agents like rodents who scavenge for food, which would be presented in the next part of the lecture.
As the SF project is about synthetic foraging, we also look at naturalistic foraging of rats in a so-called Y-maze environment. This environment consists of 3 boxes, each equipped with 3 stimulus lights. And when a stimulus lights up, the rat has to learn that he has to move over to such a light. And under this light is a reward port where he can get a fluid reward. So he must learn to navigate in this environment based on where the cue is. So in the recordings we make now from rats that perform this Y-maze task, we make again hippocampal recordings. These are each hippocampal cells. In black we have the total pathway of the rat. In yellow is the density of the firing. And this cell, for instance, fires very much at the center of the Y-maze, at the central platform. Other cells fire in an entire box, like over here and here. But some hippocampal cells fire at very small places, like only at this reward site. So the hippocampal firing is very place specific. But when we look at another structure, the ventral striatum, we see quite a different firing pattern. For instance, here this cell fires at all the 9 reward sites, but other ventral striatal cells fire much more in the center part, and they correlate in their firing to the actions that the rat undertakes at that moment. So for instance, this is the initiation of an approach response to a reward site. This is further out into the branches leading up to the reward site. And this is when the rat actually drinks the reward, the liquid. So it means that the hippocampus can be said to code for places, where am I? Whereas the ventral striatum codes much more for the action elements, appropriate for where the rat is in the task at that very moment. It's like, where am I, but what should I do here? And then you see these cells firing. And then we wondered, what is the influence of the cue? Is the cue light in any sense influencing the firing patterns? Or is it, for instance, that the hippocampus only codes for place and is insensitive to the cue? Well, the cue, as you see over here, makes a big difference, even for the hippocampal cells. When the cue is on, the rat approaches the site and crosses a place field of that hippocampal cell. And you see this as an enhanced number of spikes closely before arriving, this is a time scale, arriving at the fluid port where he gets the reward. But when he makes the same movement, but now without cue light, in the break or the pause, you see how the response of the same cell collapses. It's basically not there anymore. And over here you have a cell that barely responds when the cue light is on, but makes a strong response when the rat spontaneously approaches the same site. So this means that also the cue light is incorporated in the hippocampal system. It does have an influence on the overall encoding of memories.
So now, with this platform, we are at the point that we can actually compare the outcome, the behavior outcome of our models with the real experiments we have with the rats. So for this, we developed 2 tasks that resemble the protocols applied in Amsterdam and compare the decisions and the trajectories and this kind of behavioral information.
So what's happening here, the robot is exploring the environment, learning the world, let's say, building the representation.
And every time it stops and makes like this.
Collecting images.
He's actually collecting images and acquiring information and discovering where he is. So he's using both the information from the motors to integrate the path, and he uses this information to correct. Using the TPC, we can differentiate from different patterns. The paradigm is very easy. The robot must come here, we show a pattern, and he must go to the position in the area that is associated with this pattern. He goes, he identifies the symbol, he links to the position he must go, and he goes to the position.
Here we have an example of the representation of grid cells. And here we have a readout of the head direction cell. So it directly gives us the orientation of the robot a long time. And here we have a topological representation of place cells. So in this case, it directly gives us the position of the robot in the environment. Now that we have a model that can generate place cells in a similar way to what we found in biology, the question that still remains unclear is how to use this information to actually navigate or for foraging. First, the robot has to learn about the environment. Our proposal is that a memory system will store this information, the place cells, which will be used later on. In our memory system, we have sequences of place cells. Every sequence contains information about a goal position in the environment. By goal position, we mean food or maybe home base. Each position in this sequence contains information about a place cell, which was activated when the robot was in a certain position in the environment. So in this case, for instance, if we have a sequence of 3 elements, to keep it simple, we have a place cell here. Could happen that we have a different place here and a different one here. So if the robot has the need to go to home, it will recall these 3 different positions in the environment. So it will follow this sequence and it will go to the destination. So we can see that the robot tries to go to the destination position. When it reaches it, a different position in memory is selected and a different place cell is recalled from there. The robot has to go to reach that position. So this is an example of one sequence, but of course the memory contains a number of different sequences. But the question that still remains here is how to optimally recall this information. We can imagine that, for instance, if the robot went to home base, going around the environment, this is for sure not the optimal way to do it again. We rely in path integration, which in fact is very close to what is found in biology.